Hello everyone, Biochemia channel is here with the topic titled Carbohydrates 1. I wish it to be useful to all biochemistry lovers and do not forget to like the video to reach more people. Let's start. And firstly, let's check the contents of this topic, which the initial slides include introduction of carbohydrates, then we will do definition, and then classification. And after classification, we will start monosaccharides and their features such as formulations, projections, Fischer projection, D and L isomers, and cyclic structures of monosaccharides, Haber projection, rotation, and some physical and chemical features, including sweetness, effects of alkalis on monosaccharide, and effects of acids, reducing properties of monosaccharides, and then properties of monosaccharides related to hydroxy group they carry. Carbohydrates are the most widely distributed of the three major groups of organic matter found in living material. And so what are these three major groups? They are proteins, lipids, and carbohydrates. And in the scope of biochemistry 1, we will study all of them. We will study the structures and classification, features, and etc. And on the following biochemistry 2 course, we will look metabolism of carbohydrates, proteins, and lipids. Carbohydrates, also called sugars, not all of them are sweet, especially when they are large molecules. So in public, generally we say sugars, but the term sugar or the word sugar does not provide the definition or does not cover all carbohydrates, because we know the larger molecules or the carbohydrates having larger molecular structure are not sweet, and such as starch, which is a polysaccharide. Poly means multiple saccharides or combined to form the starch. It's not sweet, while glucose, a monosaccharide, and it is the building block of the starch, is sweet. So actually, when we say sugar in chemistry or in biochemistry, we mean monosaccharides and disaccharides, not the polysaccharides. Carbohydrates, by the way, this is the abbreviation I use, CH, capital letter CH. Carbohydrates are found in all plants and animals. And cellulose and starch is a good example. Cellulose is the most abundant in plants, which is followed by starch. While cellulose is a support and protector, starch is a food store. Glycogen has the same role in humans and animals, but the same role is about to be a food store or to be an energy source or to be a storage of glucose in body. It's not about the cellulose. So also cellulose is a good example telling us carbohydrates are more than energy. Because in general, when we talk about carbohydrates, firstly, we say or we remember the carbohydrates are the source of energy. But we will learn this is not true for all carbohydrates. Carbohydrates are more than energy, as well as the energy may be the most notable one. Carbohydrates are absorbed in the digestive system after being broken down into simplest sugars that form themselves. So this is also general for all macromolecules. For the bioavailability of the carbohydrates or for an organism, human, cat, or dog, to take the carbohydrates, proteins, or lipids from the intestinal tract, the macromolecules must be broken into their simplest structures, into their building blocks, because the macromolecular forms of carbohydrates or the other groups are not absorbed in the intestinal mucosa. The energy required for metabolism is mainly provided by the breakdown of glucose. So this is a universal molecule for all mammals. Carbohydrates are generally composed of the elements carbon, hydrogen, and oxygen. And you know the carbon and hydrogen provide the backbone of any organic molecule, and which makes carbohydrates different from the such as hydrocarbons. They carry an heteroatom oxygen. The ratio of hydrogen and oxygen atoms is generally the same as in water. Therefore, it means carbonated water. So carbonated water or hydrated carbon, also Latin for carbohydrate. However, over time, research has shown that not all carbohydrates fit this formula. So the formula is n number carbon to n number hydrogen and number oxygen, which we can simplify it as the n number carbon and n number water molecule. But I say research has shown that not all carbohydrates fit this formula. And here's an example. Look at the formula which fits to the sputtering and number carbon and the number water molecule, but this is not a carbohydrate. This is acetic acid. This is a fatty acid. This is a carboxylic acid. And look at that formula. Which it doesn't fit to this formula and number carbon and number water molecule, but it's a carbohydrate. It's a deoxy sugar. It's a monosaccharide derivative, which is rhamnose. Carbon, hydrogen, and oxygen atoms are actually present in the structure of some groups, some functional groups. Remember organic chemistry that we studied functional groups of organic molecules. And these are which are present in carbohydrates, aldehyde, ketone, primary alcohol, and secondary alcohol groups. And of these, only one of the aldehyde or only one aldehyde or ketone group is present in this structure. Both cannot be found together, and I want to put a 3-star here. This is so important. So a monosaccharide, especially the building block of carbohydrates, a monosaccharide here, actually I mean here the monosaccharide can only carry one aldehyde or a ketone group. Because for the disaccharide or the polysaccharides, they may carry both groups, depend on the monosaccharides in the structure. So this is a very critical information. What about alcoholic groups? So they complete the other carbons of carbohydrates. So actually the carbohydrates are multi-alcoholic, many of them multi-alcoholic aldehydes or multi-alcoholic ketones. Let's remember the structure of the aldehyde functional group that we studied in organic chemistry. And this is the aldehyde group. And here there is an R, a carbon backbone. And as you see, it carries an carbonic carbon and hydrogen attached to carbonic carbon. And as following the nature of the aldehyde group, the carbonic carbon is always the first carbon in this structure. So this will be important in numbering monosaccharides and also discussing their features. And here is the ketone group, a carbonyl carbon or a carbonyl group, which locates at some point of the carbon chain. So R, carbonyl carbon, and R. So, well, following the nature of the ketone group, a carbon may be at the second position, 3, or etc. But in general, in carbohydrates, actually in monosaccharides, the ketone group is the second carbon. The carbonic carbon or the ketone group is the second carbon in general. And here the secondary alcoholic group, an alcoholic group attached to a carbon, which forms two bonds with other carbons, and the primary alcohol, an alcoholic group and a hydroxyl group attached to a carbon, which forms only one single bond to another carbon. So after this information, it's time to establish a full definition of carbohydrates, or it's time to establish a definition covering all carbohydrates. So carbohydrates are defined as aldehyde or ketone derivatives of polyhydroxylic alcohols, which are monosaccharides, their polymers, the oligopolysaccharides, and their derivatives such as oxidation products, sugar acids, reduction products, which are the sugar alcohols, and substitution products, which are the amino sugars and the sulfate and phosphate esters. So this is the definition of carbohydrates. And the classification, which is there is a simple classification. Also, there is a consensus in classification of carbohydrates, which there are different types of classifications in proteins and lipids, but carbohydrates, a single type of classification, is done. So the first class is the monosaccharides, which are the building blocks of carbohydrates, and also this is the major title for today for this topic. And D and oligosaccharides, which in general compose of 2 to 10 monosaccharide units as building blocks. And if there is two, it is a disaccharide, which means 2, and from 3 to 10, they are the oligosaccharides. And there is also polysaccharides. So in general, we can say the monosaccharides, the oligosaccharides, and the polysaccharides. And polysaccharides consist of large number of monosaccharides, and their form can be a simple chain or branched complexes. And there are two classes of polysaccharides, or also I can say there are two subclasses of polysaccharides, which is the homopolysaccharides and the heteropolysaccharides. And the homo means it consists of only one certain type of monosaccharide, such as glucose. The polymerization of glucose will provide the polysaccharide. In hetero, there are two or more different type of monosaccharide units, such as glucose, fructose, Peting units. And yes, we did the classification. And let's continue. And it's time for the major title for today, the monosaccharides. Monosaccharides are aldehyde or ketone derivatives of polyhydroxy alcohols and cannot be hydrolyzed into simpler molecules. They have one aldehyde or ketone group in their structure, and as I said before, they don't carry both. And remember, these are the groups. We call them the carbonyl carbon containing functional groups, or the functional groups carrying carbonyl group. O's and a question mark. When naming a monosaccharide in Latin, the suffix "-os" is added to the end of the name. So the first thing that you should know, we use the "-os" suffix, especially for the monosaccharides and the disaccharides, because for the polysaccharides, we prefer to use their private names or their common names. So let's start with the aldose and the ketose. A monosaccharide containing an aldehyde group is an aldose, and those containing ketone groups are called ketones. So depending on the functional group they carry, we name them as aldose or ketones. So remember, my friends, there is a priority order of the functional groups, and the carbonyl group carrying functional groups always come before the other functional groups. This is why in naming we always omit hydroxyl group, especially in common names as done here. So depending on the functional group they carry, aldose and ketose, because there is no any aldoketose or a monosaccharide carrying both of them. Monosaccharides are also named according to the number of carbon atoms they contain. And for example, if it contains 3 carbon atoms, it's called the triose. So depending on the number of carbon atoms in the backbone they carry, we name them as if there is a 3 carbon, triose. 4 carbon, what about the 4 carbon? It will be tetrose. If there is a 5 carbon, pentose. 6 hexose. And let's combine both of these approaches. And a monosaccharide containing a 3 carbon and an aldehyde group is called aldotriose. Aldotriose. So the formula of the naming is first to put whether it carry aldose and ketose, then the number of carbon atoms, and put the "-ose" suffix. So if 4 carbon and an aldehyde, it will be aldotetrose. If it is a ketone group carrying 4 carbon monosaccharide, it will be ketotetrose. So we can increase examples. The most common monosaccharides in nature and in organisms are those with the 3, 5, and 6 carbon carrying. And those with 4 and 7 carbons are synthesized in intermediate metabolism or are synthesized as intermediate metabolites, and they are rare. And here is the table that you should study representing the important monosaccharides for the mammals. And here in the first column, you see the naming according to the number of carbon atoms and the carbon 3, triose, tetrose, pentose, hexose, and heptose, the naming. And in the second and the third column, according to the aldehyde or the ketone group they carry, you see the names of which are the common names actually, the names of monosaccharides. So as you see here, look at this carbon 6 carrying the hexoses. There are many hexoses. And also look at the aldose column and the hexose rows here. There are 3 aldohexoses. Aldohexoses. This means there is a difference in their molecular structure to separate them, or to call it's a glycol or it's a galactose that we will study. But before, I want to tell an additional information about each monosaccharide you see in the table. And let's start with the glyceraldehyde and the dihydroxyacetone. So they are the simplest monosaccharides. There are two simplest monosaccharides, which the glyceraldehyde is the aldotriose, and the dihydroxyacetone is the ketotriose. They have an important function in the intermediate metabolism, especially in the glucose metabolism. They occur in the glycolysis, in the breakdown of glucose reactions. And also, they are important intermediate metabolites in terms of lipid metabolism too, especially the dihydroxyacetone. In the metabolism, they are phosphated monosaccharides, or they exist in the phosphated esters such as glyceraldehyde, triphosphate, or dihydroxyacetone phosphate. And look at the carbon 4, the tetroses. We have the erythrose and the erythrulose. In general, for some monosaccharides, to express they are in the ketone carrying ones, they write the names by putting U and L before the O's to represent they are the ketosaccharides or ketone group carrying monosaccharide. And erythrose is phosphated ester, the erythrose 4-phosphate, phosphate, is an intermediate in the pentose phosphate pathway. Let me write it. Hexose monophosphate shunt, actually, correction, hexose monophosphate shunt. It is also called pentose phosphate pathway. And the erythrulose is the ketone form, is another natural keto sugar that reacts with the keratin amino acid, with amino acids in the epidermis of the skin to produce a brownish color. It reacts with amino acids, causes darkening of keratin. And ribose, which is the product of hexose monophosphate shunt. And ribose is an intermediate metabolite of the hexose monophosphate shunt, as I said, and it is required for the synthesis of nucleotides, which nucleotides are essential to produce or to synthesize the nucleic acids, DNA and RNA. And ribulose, another intermediate metabolite of, again, hexose monophosphate shunt. And as you see, many of the aldose and ketones are metabolites in hexose monophosphate shunt. And the kysylose here, the kysylose, as you see, there are two important aldopentoses, ribose and tikisylose. And tikisylose, which is Greek meaning, also Greek derivative word, which means the wood, because firstly isolated from wood, and also known as the wood sugar. It is found in the seeds of many edible plants. And the kysylitol is the alcohol of kysylose, is used as a sweetener, as a food additive. And it is involved in the heparan sulfate and chondroitin sulfate synthesis pathway, which are actually carbohydrates. And the kysylulose, again, intermediate of hexose monophosphate shunt. Glucose is an universal energy source and the most essential component of carbohydrate metabolism. Fructose, as you see, it's actually the only major ketohexose, only ketohexose. But don't forget, in nature there are other ones, but for me this is the important one. And it's also with glucose, so glucose plus fructose yields the saccharose or sucrose, table sugar. So the glucose, fructose, and the galactose, these are the main monosaccharides ingested via with foods. And also they have common pathways. So fructose also used in the glycolytic pathway as in glucose, and also it is compatible with the galactose. And galactose, like glucose and fructose, can be used as an energy source and also participates in milk sugar, which is the lactose. The glucose plus galactose will give lactose. And also the galactose may be the most notable significance of the galactose is to be a structural component of membrane lipids, especially in nerve tissue. And mannose, another aldohexose, like in the glucose and fructose, a component of glucose metabolism, and also the other function, maybe the notable one for the mannose, it is involved in the glycosylation of some proteins, especially it can be a component of, it is the component of some oligosaccharides, which they are conjugated with the protein. And we call it the glycosylation of proteins. And pseudoheptulose, one of the few heptoses, and you see it's a ketoheptose in nature. It is found in various fruits and vegetables, such as carrots, apricots, apples, and even in tomatoes. And it's an intermediate metabolite of hexose monophosphate shunt, as seen as we say also cellulose, and the other ones. And also some studies show that it reduces pro-inflammatory markers. And for more monosaccharides, if you want to learn the more monosaccharides, you can visit my website, biokimia.web, and search for an article titled The Smallest Building Units of Carbohydrates, the Monosaccharides. And there you will see the other ones, such as, for example, allose, glucose, talose, interesting names. But for the cost, these are okay. Formulation of monosaccharides, and the first one is the closed formulas. And look at here. There is a formula carrying 6 carbon, 12 hydrogen, and the 6 oxygen. And I said, as a given information, as a correct data, it's a carbohydrate. So the question, by looking at the formula here, what can you say about this monosaccharide? And there's no possibility to say whether it's an aldehyde or ketone carrying, or also the number of alcoholic groups. But if we are sure about it's an monosaccharide, we can only say it's an hexose because it's clear that it carries 6 carbon atoms, which makes it an hexose. But no possibility to say whether it's remember the hexoses from the previous slide. It may be glucose, galactose, mannose, or fructose. So by checking this formula, we cannot make a differentiation. And look at here, a more open formula. And so C and the carbon to hydrogen, and then there's alcoholic group CHOH. There's a 4, the CHO. So by checking this formula, and from the previous one, we know it's an hexose. We know it's an hexose. Again, we can say it's an hexose because there are 6 carbon atoms, 6 carbon atom. But here, this time, we can say more. And look at here, CHO. So this is the pattern for the aldehyde, and here this is the carbon backbone, the R. Now we can say it's an aldehyde carrying carbohydrate. So it's an aldehexose. And also by checking the formula, we can say there are 4 secondary alcoholic groups, and here the 1 primary alcoholic groups. But remember the previous slide, remember the number of aldehexoses we have, glucose, mannose, and galactose. Still, by looking this formula, we cannot detect whether it's a glucose, mannose, or galactose. Structure formulas of monosaccharides. It's a form that shows the space alignment of the groups. And the carbon atom carrying the aldehyde group in an aldehyde is defined as the first carbon atom. So this is general feature of aldehydes. And the carbon atom carrying the primary alcoholic group is defined as the cis carbon. Also, this is natural. In aldoses, there are only 1 primary alcoholic group, but in ketone, there are 2 primary alcoholic groups. And a similar approach also done for the ketones, which we firstly write the carbon atom, the carrying the ketone group at the upside of the 2-dimensional plane. And here, firstly, we're from up to bottom, the carbon atoms. If it's an aldehexose, there are 6 carbon atoms. And then at the first carbon, at the upper side, is the aldehyde group, which is the first carbon, and following second, third, fourth, and fifth carbons are the secondary alcoholic groups, and the sixth carbon is the primary alcohol group. But remember, there are three aldohexoses: glucose, mannose, and galactose. Still, we need more information because the formula you see or the given formula is not clear which one is defined. We can only say again it's an aldohexose. So to determine the type of which aldohexose we have, we need to check the configuration formulas. And it is the shape showing the positions of hydrogen and hydroxy groups by the carbon atom in the structuring space. And many carbohydrates can be formed according to the arrangement of hydrogen and hydroxy groups in space. And look at here, this is the glucose. And compare this given formula with the previous one. And focus on the third carbon. And look at here. Focus on the third carbon. The hydroxy group is at the left side. And let's check the other example, the fructose, which is a ketohexose. And here, let's highlight the functional groups, aldehyde and the ketones. And again, look at the third carbon at the left side. So firstly, we have to identify the aldehyde or the ketone groups. Then we should check the spatial arrangement of the hydrogen and hydroxy group, whether they are at the left side or the right side. So this means by this arrangement, several monosaccharides may form. Fischer projection is a projection to make understandable for the configuration formulas of monosaccharides. And it is established by Hermann Emilius Fischer, a German chemist and the winner of the Nobel Prize in Chemistry in 1902. He discovered Fischer esterification and also developed the Fischer projection, a symbolic way of drawing asymmetric carbon atoms. Remember asymmetry from organic chemistry. So this projection is not only for the carbohydrates, but for all organic chemistry, all organic compounds especially showing asymmetry. And now also we will study the asymmetry of carbon atoms in the monosaccharide. He also postulated the lock and key mechanism of enzyme action. And as a footnote, and additional information about him, he never used his first name and was known throughout his life only as Emil Fischer. According to this projection, in the vertical plane, carbon 1 is drawn at the top of carbon chain and carbon 6 at the bottom if it is hexose. And also same for pentose. In an aldose, the carbon of the aldehyde group is carbon 1. And in a ketose, the carbon of the ketone group has the lowest possible number, and usually carbon 2. In this project, the L and D structures, actually the isomers, L and D isomers, are also distinguished. It is used to represent mainly monosaccharides, but also amino acids, and I said organic compounds. Especially in the demonstration of enantiomers of the chiral molecules in stereochemistry, we need to draw their Fischer projection. And let's remember the asymmetric carbon atom. In general, a molecule with n chiral centers can have up to 2 to power n number of stereoisomers. Recall that in stereochemistry, stereoisomerism or spatial isomerism, which is a form of isomerism in which molecules have the same molecular formula and bonded atomic sequence or structure, but three-dimensional orientations of the atoms in space are different. And it was the chiral carbons that determine this. And remember the chiral carbon, a carbon, and it must be in sp3 hybrid model and forms four single bond with different atoms or group of atoms as X, Y, Z, and alpha. If this pattern exists in a carbon atom, we call it a chiral carbon or asymmetric carbon or stereogenic center. Recall from organic chemistry this information. And also I wrote here the n number chiral carbons or chiral centers means number of asymmetric carbon. And for example, the glyceraldehyde, let me erase this. Look at the glyceraldehyde and the dihydroxyacetone. They are the simplest monosaccharides, which each carry three carbon. And glyceraldehyde is an aldehyde, and dihydroxyacetone is a ketone. And in dihydroxyacetone, there are two primary alcoholic groups, but there is no any secondary alcohol group. And when you look at the carbon, the first carbon, there are two hydrogen. This means this is symmetric carbon. This is not a chiral center. This also not a chiral center, and this also not a chiral center. Let's look at the glyceraldehyde. And the carbonic carbon is not a chiral center. And also in the primary alcoholic group, there was two hydrogen, which makes this carbon symmetric. Thus, this is not a chiral. But look at this one. We have an X. This group, as I said, group of atoms, the Y, this is the Z, and this is the alpha. So there are four different groups or group of atoms attached to this carbon via a single bond. And so this is in sp3 hybrid model. So this is the chiral center. So for the glyceraldehyde, the num n is 1, thus there are two isomers of glyceraldehyde. And remember, if an organic compound having one chiral center, its isomer is the mirror image. It's enantiomer. It is enantiomerism. For the ones carrying two or more, it may be there may be enantiomers but in general they are the diastomers. Yes, you see the Fischer projection of galactose, mannose, and glucose. Now it's time to differentiate these three aldohexoses. And as you see, both are carrying the aldehyde, which makes them aldose, and all carry 6 carbon atoms, so they are aldohexoses. So what is the difference? Focus on the third carbon. All carry an hydroxyl group at the third carbon in the left side, which means the same position. But what about mannose? What is the additional feature of mannose? It has a hydroxyl group at the left side at the second carbon position. This is the difference. So, an approach to learn it, so draw a single vertical line representing the 6 carbon backbone. And for the glucose here, draw a horizontal line and put the 3. So this is the glucose, a single backbone, and at the 3 point, left horizontal line represents this hydroxyl group. So put the rest. And for the mannose and the galactose, also let me delete and draw again for the better, and also to match the position. Yes, here, the third position. This is same for all, as you see. In a mannose, there's an additional hydroxyl. An additional feature in mannose is the hydroxyl group. The second carbon is also at the left side. So 2, 3. This is mannose. For the galactose, we get here 3, 4. So this is the difference. And when we compare the glucose and mannose, we say glucose and mannose are epimers. And mannose and galactose are not epimers, but glucose and galactose are epimers. So a new feature for you. In stereochemistry, an epimer is one of a pair of diastomers. The two epimers have opposite configuration at only one stereogenic center out of at last two. This means a monosaccharide that differs from another due to difference in a spatial position of the one hydroxyl group is called epimer. And all other stereogenic centers in the molecule are the same in it. And also epimerization, which is the process of interconversion of one epimer to the other epimer can occur. So when we compare the glucose and mannose, there's only one difference in spatial arrangement. This is why glucose and mannose are epimers. And when we compare glucose and galactose again, there's a difference in the one position. So there are the epimers, but when we look at the mannose and galactose, two are in the left side. This is why galactose and mannose are not epimers. After explaining the epimerization or the epimers, which the galactose and glucose are epimers and the galactose and mannose are epimers, it's time to talk about the isomers, D and L isomers. Monosaccharides with different lengths of carbon chains have two groups of stereoisomers, and these groups differ in the configuration about the chiral center that is farthest from the carbonyl carbon. So this is important. These groups differ in the configuration about the chiral center that is farthest from the carbonyl carbon. The stereoisomers that have the same configuration as D-glyceraldehyde at the reference carbon are called D-isomers. And those identical to L-glyceride are known as L-isomers. And let's look at the D-glyceraldehyde and the L-glyceride. So, put simply, when the hydroxyl group is positioned to the right of the reference carbon in a projection, where the carbonyl carbon is at the top, as you see in here, the sugar is classified as the D isomer. And so this is the hydroxyl group, which is farthest to the carbonyl carbon in D glyceraldehyde. So this is the D isomer. And here the D refers dextro, which means right, but this is Latin. When the hydroxyl group positioned to the left as seen here, is the L isomer. And here the L means level. In Latin, this is left. And for example, for the Fischer projection of glucose, remember the formula of the glucose, which carries 6 carbon, and for chiral center in the Fischer projection of the chain structure, which makes the 2 to power of 4, 16 stereoisomers, which means the 8 in D forms and 8 in L forms. But the majority of the living organisms use only D isomers for the metabolic purposes. Although the reason for this is still a fascinating, and also there is no clear answer about the question why D forms of or the D isomers of monosaccharides are used in metabolism. And also the same isomerism seen in amino acids. Also there are D and L isomers of amino acids. And interestingly, the L form of amino acids can be used in metabolism. Even we don't have any clear answer, especially the why in the initial or the early stages of evolution this happens, we know that the enzymes may be the answer because enzymes in metabolism for the monosaccharides mainly catalyze the D forms, or the enzyme has the active sites which can be used D isomers for monosaccharides as a substrate. But for the amino acids, L isomers. Let's solve a problem using the reference molecule glyceraldehyde, and it's in D isomer. So look at the hydroxyl group attached to the chiral carbon, which is the farthest chiral carbon to the carbonyl carbon, and it's in D form. And let's determine the given glucose molecule is in D form or L form. And the definition tells us determine the farthest chiral carbon to the carbonyl carbon. This is the carbonyl carbon, and this is a chiral center, and this one, and also the fifth carbon is the chiral center. So which makes the fifth carbon the farthest carbon to the carbonyl carbon. And look at the position of the hydroxyl group, which is same with the diglyceraldehyde, which makes the glucose molecule the D isomer. And here is the L-glyceraldehyde, and look at the position of the hydroxyl group attached to the carbonyl carbon, attached to the chiral center, chiral center, the farthest to the carbonyl carbon. And look at this molecule here, the given molecule, the glucose molecule. So we have the carbonyl carbon at the first position, and the second, third, fourth, and fifth carbons are the chiral centers, which the fifth one is the farthest one to the carbonyl carbon. And as you see, the position is same with the L-glyceraldehyde, which makes the glucose L-glucose. So, in addition, here, if you realize, there are plus and minus signs into the parentheses. And these are refer the optical isomerism. So D and L forms are also optical isomers, or they are optically active molecules, which means they scatter the polarized light. And if they scatter to the left side, we call them levorotatory, and they are referred as the minus sign in the parentheses, and the plus means dexarotatory, which they convert polarized light to, which they divert polarized light to the right side, and thus we use the positive sign into the parentheses to refer they are dextrarotatory. For simplicity, we have represented the aldohexoses or ketohexases structures as straight chains. So we have a Fisher projection chain. But also we know, especially in aqueous solution, aldoteteroses and all monosaccharides with 5 or more carbon atoms, such as 6 and 7, in the backbone occur predominantly as cyclic structures, ring structures. in which the carbonyl group forms a covalent bond with the oxygen of a hydroxyl group along the chain. In shortly, there are cyclic structures for the hexoses and heptoses, and also aldotetroses. So for the hexose and heptose, both aldohexose and ketohexoses, or both forms. But for the tetroses, only aldotetroses. And this happens in aqueous solutions. The formation of these ring structure is the result of a general reaction between alcohols and aldehydes or ketones to form derivatives called hemiacetals or hemiketals. And so for the topic, please remember the organic chemistry functional groups, the aldehydes and the ketones. So you may turn back to organic chemistry to check your notes. In two molecules of an alcohol can be attached to a carbonyl carbon. And so the product of the first addition is a hemi structure, hemiacetal or hemiketal. And here, if the hydroxyl group and the carbonyl carbon comes from the same molecule as seen in monosaccharides, a 5 or 6-membered ring is formed. If there is an addition of the second alcohol group, so it will produce a full acetal or the ketal group, and the bond formed, if it is a monosaccharide, is the glycosidic bond. So in formation of the saccharides, two monosaccharides form the glycosidic bond, and here one of the monosaccharide or maybe the two will form the full acetal or ketal structure to yield the saccharides. As the ring shapes are formed, such as the reaction with the first alcohol molecule creates an additional chiral center. So to production or the formation of the hemiacetal or hemiketal structure also means the formation of an additional chiral center, which will, for the aldohexoses, which will produce the fifth asymmetric carbon atom or the fifth chiral center. And isomeric forms of monosaccharides that differ only in the configuration with respect to the hemiacetal or hemiketal carbon atom are called anomers. And the carbonyl carbon is called the anomeric carbon. And the anomeric carbon is the fifth asymmetric carbon in an aldohexose, or in an aldopentose, it will be the fourth asymmetric carbon atom. And since the alcohol can be added in one of two ways, either front or the back of the carbonyl carbon or in a two-dimensional plane, left or right of the carbonyl carbon, so the reaction can produce one of two stereoisomeric configuration, which we define as alpha and beta forms. Or for the anomers, there is a two possibility, which is the alpha anomer or the beta anomer. So the formation of the ring will produce fifth asymmetric carbon atom, which is an anomeric carbon, and due to the possibility of adding hydroxyl group into left or right or front or back of the carbonyl carbon, we have an alpha and beta anomeric monosaccharides, or the alpha and beta anomers of monosaccharide. So this is the phenomena of the formation of the hemiacetal and the hemiketal structure. And these isomeric forms of monosaccharides that differ only their configuration with respect to the hemiacetal or hemiketal carbon, we call them anomers. Don't forget, we call them anomers. So it is in the right, I said alpha anomer. If it is on the left side, we call it the beta anomer. And for the anomer, or the type of the anomer, which is determined by, determined by the hydroxyl group, which will be also a free hydroxyl group, or in total we call it the hemiacetal or hemiketal group. It also provides a reducing property to the monosaccharides. If there's a free group, free hemiacetal or ketal, or sometimes we call the free hydroxyl group in the anomeric carbon, it has a reducing feature. Yes, during the formation of the cyclic structures, we know there is a two possibility to occurrence. The first possibility is the occurrence of the five-membered ring and the six-membered ring. And let's define these ring structures. So the compounds with a six-membered ring are called the pyranose structure. So the O refers the monosaccharide, the pyran refers it's in the pyran or it's in the six-membered ring structure. Why we call it pyran? Because they resemble the pyran. And for the ones which have the five-member ring, we call them furan ring, or shortly they are called furanos. Furanos. And look at the ring. This is the pyran ring. This is the pyran ring. So if there's a molecular shape similar to pyran, we call them the pyranos. And thus, if we have a glucose molecule in the cyclic structure of pyran, we call them in a chain structure, it is glucose, such as in the D-glucose. If it is in the ring structure, or the name will be the glucopyranose. Glucopyranose. So D-glucose means refer its chain structure. If you want to refer it in the aqueous solution as in the ring structure, it is pyranose. And also there's the possibility for the glucose to be in the five-member ring. Five-member ring. And this is furan ring. Furan ring. So if the cyclic monosaccharide has a similar structure with the furan, it will be furanose. Furanose. And then the glucose, for example, D-glucofuranose, or mostly in ketos, especially in ketohexoses, and it's a fructose, it will be as a fructofuranose structure, D-fructofuranose or D-glucofuranose. If it's in the pyran structure, D-glucopyranose, or mannose, for example, D-mannopyranose or galactopyranose, if you talk about the galactose. So in an aqueous solution, both for a given monosaccharide, for a given aldohexose or ketohexose, both pyran and furan rings can exist, as well as the chain structure. For example, in a glucose, in an aqueous solution of glucose, the dominant form is the pyranose. But for the fructose, about 67% in pyranose and 33 in furanose. And for the ribose, 75% furanose and 25% in pyranose form. And for better understand these structures, we should study Hubble projection. And yes, Hubble projection, which is identified by or discovered by Sir Walter Norman Howard. And these cyclic structures are discovered by the Hubble projection. Also, it set or he established the rules to present the cyclic structures of the monosaccharide. And Sir Walter Norman Howard, he was a British chemist known for his groundbreaking work on ascorbic acid, which is vitamin C, and also the explanation or the projection of cyclic structures, especially three-dimensional cyclic structures of monosaccharides, or in true words, he established the rules to translate the three-dimensional structure of monosaccharides into convenient two-dimensional graphical form. And in 1937, he was awarded the Nobel Prize in Chemistry for his contribution to the vitamin C and also this projection. Yes, according to the Hubble projection, the cyclic structure of monosaccharides are present at fifth and sixth carbon members. And carbon is the implicit type of atom, and carbon 1 is known as the anomeric carbon. Hydrogen atoms and carbon are implicit. A thicker line indicates atoms that are closer to the observer, and sometimes we can omit. The groups below the plane of the ring in Hubble projection correspond to those on the right-hand side of the Fisher projection. This means, think about this is a Fisher projection of the glucose. So if we turn molecule to the clockwise at the bottom or at the 90-degree angle, so the hydroxyl group, which is at the left side in Fisher projection, will be the upper position. So this explains, or this is an example why I wrote here the groups below the plane of the ring in Hubble projections correspond to those on the right hand side of the Fisher projection. And for better understanding, first of all, I want to express how anomeric carbon occurs here. So compare the D-glucose. This is in Fisher projection, or the glucose structure, the glucose straight chain structure, which there is no cyclic structure seen here. And these are the beta D-glucose and the alpha D-glucose. These are the two possible anomeric forms of glucose. And according to the explanation of the cyclic structures, what I said, the first hydroxyl group here forms an oxygen bridge with the carbonic. Actually, this a transfer of hydroxyl group to this carbon, which makes a convenient space to occur the oxygen bridge. So this is transferring of alcoholic group to the carbonic carbon. So this is an hemiacetal structure. If we add one more hydroxyl group to this carbonic carbon, it will be a full acetal structure. And so the same thing happens here, but focus on the sides of the hydroxyl group. So there are two possibilities, which the beta one is common. Beta one is the common in an aqueous solution, mostly in beta form. And so if it's the right side, this is the alpha ionomer. If it's the left side, it's in the beta ionomer. And what I said, if we turn the molecule in the clockwise, this means the left side will be in the upper part of the molecule, and the right side will be the bottom of the molecule. And here, actually, the real representation of Howard projection, and the molecule which the groups close to us are represented as you see in here, thick lines, and the part of the molecule located towards us represent in the thin lines. And fifth and the first carbon here, which forms the oxygen bridge, thus this is the anomeric carbon. And this is in the upper part, which means when we rotate the clockwise, if open the ring and clockwise, anti-clockwise, or sorry, counterclockwise rotation. So it will be left. So this is the beta ionomer. And be careful. This is the first. To determine which one is the first, actually here it is given to you. but maybe it's not given to you. So you can easily determine, focus on the oxygen here and the position of the atoms close to the oxygen. And then you can easily identify this is the alpha atom because here fifth and one more carbon here, which is the sixth carbon, and also count 1, 2, 3, 4, 5, then six carbon carrier. So here it's in the bottom, which means at the right side. So this is the alpha D glucose, and this is beta D glucose. So how we determine the glucose? Remember the Fischer projection. We have the hydroxyl at the third carbon atom on the left side, which will make it to be at the upper part of the molecule. And look at here, this is right in Fischer, but look at here, the third position, this is the upper side. And in general, also what I said, we can omit to represent the hydrogens atom. We can implicit, and also here the carbons are implicit. But we know at each point here, there is a carbon atom. But here, this is oxygen bridge. And look at the structure, it's similar to the pyron ring. Actually, this is the form beta D-glucopyranose. Sometimes we omit, but we know it's in pyranose, glucopyranose. And in an aqueous solution, this is the predominant form or the dominant form. And so also, this is another good example how the pyranose hemiacetal structure of the beta D glucose located its upper part, which means the left here when occurred, and this is the hemiacetal structure of the beta D glucopyranose. And actually, it's also a kind of ring chain tautomerism of the glucose, the pyranose form. So the conversion between the open and closed forms of a molecule is called the ring chain tautomerism. So this means there may be a interconversion between the anomeric forms to the chain structure form, and it is called tautomerism, ring to chain interconversion, tautomerism, ring chain tautomerism. Another title, mutarotation, a title regarding the anomers. The alpha and beta anomers of D-glucose, are interconverted in aqueous solution by a process called mutarotation. In this process, a ring form, such as the alpha anomer, opens briefly to the linear form, then closes again to produce the beta anomer. Thus, a beta D-glucose solution and an alpha D-glucose solution eventually form identical equilibrium mixtures with the same optical properties. And the mixture consists of approximately 1/3 alpha D-glucose and 2/3 beta D-glucose and very small amounts of linear and linear forms. So all forms exist in a given aqueous solution. And here the D-glucose and the alpha beta D-glucose and then the alpha D-glucose. And also this is explained by the rotation of the polarized light. And as you see, each rotate the polarized light, polarized light into same direction. All are in dextrorotatory, but in different angles, And so these molecules are optically active. So they are stereoisomers. They have chiral centers. And don't forget about the diastereomerism or the information in stereoisomer. What we said, if a given organic molecule carry two or more chiral centers, it can be optically active or not. And the monosaccharides are the aldohexoses and the ketohexose are optically active compounds. And we can determine the rotation of light by using the polarimeters. Sweetness of monosaccharides. Sugars are sweet substances. So the sugar means monosaccharides and saccharides. And the sucrose is known as the benchmark for sweetness, rated at 100, while other sugars are rated comparatively. And so, what is the source of the sweetness? It's about the alcohol groups they carry. The term sugar is derived directly from their taste. In reality, what we commonly call sugar is the sucrose, also known as table sugar or granulated sugar. And this is a type of the saccharide, not the monosaccharide. And here, a table comparing the sweetness of different monosaccharides and the disaccharides. And here, the lactose, maltose, and the sucrose. These are the disaccharides, and the rest are the monosaccharides. And the galactose, glucose, fructose, they are hexoses, and the ketosis is the pentoses. And look at the one which has the highest sweetness is the fructose. And the sucrose is the 100, so the rest are lesser sweet comparing to the sucrose and fructose. Effects of alkalis on monosaccharides. They dissolve easily in water and aqueous media. The way in which they react and behave differs depending on the level of the acidity, alkalinity, or neutrality of the liquid they dissolve in. They are also soluble in ethanol, but insoluble in the ethyl ether. In water and neutral aqueous media, they show a slow mutarotation. In very slightly alkaline media, mutarotation accelerates. And in slightly alkaline liquids, such as 0.05 normal concentration or equivalent concentration, certain types of monosaccharides undergo a transformation into different enol forms. And the process is called enolization. Enolization is a process in aldehydes and ketones where a proton migrates from a carbon atom to the oxygen of an adjacent carbonic group, aldehyde or ketone, to produce an unsaturated alcohol called enol. And the resulting products are known as NODOs because at the end, during analyzation of the hexoses, there will be a two alcoholic group attached to the carbon-carbon double bond. So remember the enol. If there is a carbon-carbon double bond and in which the one carries an alcoholic group, this is called enol. If both carry alcoholic group, it will be a enodiol. In strongly alkaline environments, the reaction abilities of enediol forms significantly increase. In concentrated alkaline environments, when monosaccharide solutions are boiled after being thoroughly mixed, color from yellow to red and at the end brown can be observed. And this color change results in the formation of resin-like substances, and the smell of caramel is also present during this process. Yes. So here you see the mannose, fructose, and the glucose, and so they can convert to each other via the enodiol intermediate. And so look at the carbon backbone from third carbon to the sixth carbon. They carry the same structure. And the difference is on the first and second carbon, first and the second carbon. And so from the mannose, the replacement or the rearrangement from between the 1 and 2, the proton move here and the double bond move here. So a carbon-carbon double bond carrying two hydroxyl groups, and this is the enodiol structure. And for the fructose, also this move to here to form the enodiol structure, or vice versa. And in glucose, the hydrogen moves to here to form the enodiol structure. And during this conversion, a color change is occurred in alkaline solution, which is the Moore experiment. It is based on the formation of reductive, especially the yellow colored enol groups, which the enol dual intermediate, and there is an rearrangement between carbon atoms 1 and second. And here, then this event occurs in a temperature in an, or the influence of heat in alkaline environment. Effect of acids on monosaccharides. In acidic conditions, monosaccharides undergo accelerated metabolization as the initial reaction. If the temperature is raised to boiling in highly acidic environments, monosaccharides are carved. And some aromatic compounds produce specific color reactions with monosaccharides, and we can use them for identifying some of them. And boiling monosaccharide molecules in concentrated acidic condition results in dehydration causing the loss of 3 moles of water. And this creates furfural derivatives or furfural from pentoses and 5-hydroxymethyl furfural from hexoses. Main characteristics of furfural and 5-hydroxymethyl furfural derivatives, they create color reactions by combining with some aromatic compounds such as ocene, fluoroglossinol, resorcinol, and alpha-naphthol. In the Molish experiment, which used alpha-naphthol, yields a violet color. It's a general test for recognizing sugars. All sugars, whether free or bound to protein, give this reaction positive. Therefore, the Molish is the general recognition reaction for sugars, with a few exceptions. The Selivanov's test reaction using resorcinol produces a red-pink color and is specific for ketohexoses. Ketohexoses are less resistant to the action of hydrochloric acid in high temperature than the aldohexoses, and they react with resorcinol, which is a 1,3-dihydroxybenzene, and as a common feature, all are aromatic compounds which they carry benzene, which forms and to give hydroxyfurfural derivatives, which forms a characteristic red complex. And pentoses react with ocene to produce a green color and with fluoroglycinol to produce red color, aiding the identification of pentose. So for Molish, I said it's a general test to recognize the presence of a sugar. Selivanov for the ketohexose and for the pentose orcine or fluoroglycinol. And the orcine, we have the experiment Bial experiment or the Bial's test. Reducing properties of monosaccharides. Monosaccharides and some disaccharides are strong reducing agents, especially at high pH. This means in alkaline environment. And these properties are due to free aldehyde and the ketone groups they carry. They easily reduce ions such as silver, mercury, bismuth, and copper. And in the meantime, they themselves oxidize and form acid. And if these reactions are carried out with the addition of a colored oxidizing agent, a color change will occur when the monosaccharides are reduced by the monosaccharides and the reduction will become visible. And the free groups, aldehyde or ketone, or in cyclic structure, remember, in an aqueous solution, the dominant form is cyclic, and in cyclic structure, they carry an hydroxyl group to the anomeric carbon, or shortly we can say the free hemiacetal in aldehyde and the hemiketal in the ketoses. They undergo the redox reaction. They reduce and itself oxidize. And the common principle of these experiments regarding or relying on the reducing properties Sugars with free aldehyde or ketone group show the ability to reduce these heavy metals. And during the experiment, the sugars oxidize and give sugar acid, aldonic acid. And also they give color reactions to make them visible. And these experiments are the Fehling, a famous one, and which was formerly used for the quantification of glucose in blood too, and the Benedict experiment and the Barford experiment. So they have the almost same principle. And properties of monosaccharides related to the hydroxyl group or alcoholic group they carry, and which one is the glycoside formation. When the ring structures of monosaccharides are combined with alcohol or phenyl groups using hydrochloric acid as a catalyst, glycosides are formed. And the bond formed between the aldehyde group of one monosaccharide and the alcohol group of another monosaccharide to form a glycoside is called a glycosidic bond or glycosidic linkage. So, and this linkage also is a type of ether bond that joins the carbohydrate molecule to another group, which may or may not be another carbohydrate. And the glycosidic bond is formed between the hemiacetal or hemiketal group of a monosaccharide and the hydroxyl group of some compound such as an alcohol. And the substance containing a glycosidic bond is called glycoside. And the glycoside term is now extended to also cover compounds with bonds formed between hemiacetal or hemiketal groups of sugars and the several chemical groups other than hydroxyl, such as the thioether, which give thioglycosides, selenium, selenoglycosides, and the other ones. And most naturally glycosides or natural glycosides are found in plants. For example, salicylin, aspirin-related glycoside, oleandrin, cardiac glycoside. And particularly in naturally occurring glycosides, the compound hydroxyl or the carbon backbone to hydroxyl from which the carbohydrate residue has been removed is often termed the aglycon. And the carbohydrate residue itself is sometimes referred to as the glycon. And ether formation, they are formed as a result of the replacement of hydrogen in hydroxyl groups with alkyl groups. And also it is related with the glycosidic linkage formation between two, such as between two monosaccharides. In ester formation, sugar esters are formed by esterification of hydroxyl groups with acids. And phosphate esters created using phosphoric acid hold paramount importance in biology. They participate in the carbohydrate metabolism and the composition of nucleic acids. And here some examples are given in the table. The two esters, the phosphate esters of glucose, which one is the glucose 1-phosphate and 6-phosphate, the other one. And for the fructose, fructose 6-phosphate and fructose 1,6-biphosphate. Harden-Young ester, Nebus ester, Robinson and the Corey ester. And they are so important in the metabolism. And the common feature in carbohydrate metabolism, all intermediate metabolites, which some are given here, are the phosphate esters or phosphate derivatives of monosaccharides. And an esterification of a monosaccharide phosphate is a kind of activation or is a kind of delivering the monosaccharide for further metabolic reactions. It's so important. And also look at the ATP, which is the energy molecule, is an adenosine triphosphate, and the adenosine moiety carries a ribose 5-phosphate, and this is 5-phosphate. This is the component of any nucleotide, including DNA and RNA. And my dear friends, to finish the topic, and here are the references for you, both in Turkish and English. You can get any of them for further studying. And the next topic will be carbohydrates 2. Also, we say part 2. And for more about the biochemistry, clinical biochemistry, and also the laboratory world, you can visit my webpage, biokimia.vet. You can also follow my Instagram account for up-to-date information or any shares about the biochemistry, lectures, etc. And please don't forget to subscribe to your channel, click on thumbs up and open notification for further news. See you in our next topic. Goodbye. Stay safe.